go?
You wrote.
Okay. Hello, everyone. I'm Dina Drennan. I work in the Geo and Environmental Microbiology lab here at CSM under Josh Sharp. I spend a lot of my time thinking about sulfate-reducing bioreactors and treating mining impacted water, specifically the microbial ecology and geochemistry thereof. I would like to introduce Dr. Alex Burgin to speak with us today about small molecule modulators of protein-protein interactions. Dr. Burgin received his PhD in genetics at Indiana University in 1990 and was one of Normposium's students on the RNA-SP project. He went on to complete postdoctoral studies at the National Institute of Health, and following that, he was senior scientist and group leader at Ribozyme Pharmaceuticals. Dr. Burgin joined the faculty at San Diego State University in 1998 as an assistant professor in the biology department. And in 2001, he joined Emerald Biostructures as director of molecular biology. Since 2013, Dr. Burgin has been working at the Broad Institute of Harvard and MIT as the senior scientific advisor to the CSO. And his research includes better informing the molecular pharmacology of Alzheimer's disease treatment. With that, please join me in welcoming Dr. Burgin to the Normposium at the Colorado School of Medicine.
It's a great honor to be here. This is really fantastic. I'm going to tell you a story that's going to be a little bit different, but I think that's what's so great about this Normposium. We're getting lots of different kinds of talks. As was mentioned, I was a graduate student in Norm's lab from 1986 to 1990. I worked on the P project, so I was on the biochemistry side of the lab. And you're going to wonder, how did I get some RNA to protein-protein interactions? And hopefully, what I'm really going to try and tell you is how Norm made me a better drug hunter. There's a lot of lessons here. One of the first ones, and so I started formulating this talk, I realized that there's so much I got from Norm's lab that I didn't really appreciate until I actually went back and tried to consciously think about it. As I started thinking about it, it didn't take me very long. There are several things. One of the first things that has lasted with me and stayed with me, and I think has been fundamental to my success as a scientist, is he really showed me to always think, has nature already done the experiment? I remember being in the lab, suggesting something, and Norm very quickly saying, well, nature's already done that experiment. And there's so many corollaries to this. We've already talked about how things that are conserved are important, and there's so many corollaries to this basic idea, and it's such an important one that I don't think is really appreciated very often. And hopefully I'll show you some of the examples of how I've been thinking about that and how that's influenced my work. Another one that I hate to admit after being a graduate student during those RNA processing days is enzyme kinetics are really important and really useful. It's painful as a graduate student to learn them, and it's actually really disheartening to me as I've gone now decades later and see how the rigor and the amount of appreciation for enzyme kinetics really wanes, and it's actually really, I think, a real problem in modern drug discovery. Third thing, structure informs function. I learned very early in Norm's lab, sort of took it for granted, it was so obvious. Of course, if you want to understand how RNA-SP works, you got to understand the structure. And it's amazing to me when people are making drugs today, so little effort is actually paid to understand the structure of the target and how the small molecules might be modulating that structure. And then finally, I think one of the most important things, and this I did recognize in Norm's lab, be courageous. One of the reasons why I chose Norm's lab was because it was so impressive to find someone who had the courage to be doing microbial ecology and hardcore RNA biochemistry. And once I started my own lab, I realized how difficult that really was. And I've tried to always live by that motto. I think it does take a lot of courage to not be afraid, okay, I need something, that means I have to get in the hood and actually chemically make it. So I need to learn synthetic organic chemistry. Or I have a new molecule, I understand how it works, and so I need to get into the laboratory and understand how do we deliver it to a rat, and what does the rat do? I mean, to be a great scientist, you have to have, I think, a tremendous amount of courage. And it's remarkable to me, that's the one thing that I think is so impressive about Norm. The fearlessness to answer the question and go after the problem. So, here's the story I'm going to tell you about. I'm going to tell you about 10 years ago, I had the opportunity to lead a project or drug discovery project to find inhibitors of PDE4 for the treatment of cognition. And this is a great opportunity. When I first had this, we stepped back for a while and thought about the problem. And this is a really big problem. It was kind of a daunting one because PDEs are phosphodiesterase. They catalyze the hydrolysis of cyclic AMP or cyclic GMP into the 5' phosphate cognate. And of course, those are critical cyclic messengers that are involved in all kinds of cellular processes. And so the drug companies have been interested in these enzymes for many, many decades. And they've gone after many different forms of the enzymes. There are actually 11 different superfamily members. People have been actively trying to make drugs against this set. You might know PDE5 as Viagra or Cialis, the erectile dysfunction. That's the only PDE that's been successfully drugged. The one that has critically had the most effort, most attention is PDE4. This is the primary enzyme which catalyzes the hydrolysis of cyclic AMP in our cells. And people for decades, literally since the late 60s, have been going after this target for asthma, depression, a variety of inflammatory diseases. We had the idea we wanted to go after cognition. And we knew it was going to be a big problem because if you look at why this family has been so difficult to drug, is that there are 11 superfamily members, and you can see the unrooted tree here showing the crystal structures of all the catalytic domains. Those catalytic domains, not surprisingly, are highly conserved. And so it's fairly easy to make, well, I shouldn't say easy, it's possible to make something which targets PDE5 because it's hydrolyzed cyclic GMP. But try to imagine targeting PDE4, which has 4 different genes, which have very different biological functions, but are absolutely highly conserved. You can see at this root of this tree how closely related, and structurally you can hopefully see the active sites are identical. If you make a drug that targets PDE4D, it'll inhibit PDE4B, and vice versa. And that has resulted in lots of problems. And so for decades, people have tried to do this. They've been focusing on active site inhibitors, and they fail. They have dose-limiting toxicities in clinical trials, all the way back into the 70s when Rolopram was first studied for depression. You get bad nausea, you get vomiting, you get diarrhea, you can't keep the drug in you, and it fails. Also, in preclinical trials, it's really kind of scary because it's very easy to kill rats with PDE4 inhibitors. You get these horrible, it's unique to rats, but still, it's scary when you think about trying to get a drug through the FDA. How are you going to convince someone, yes, it kills rats at really low doses, but you should go ahead and take it. And the death is really horrible because it causes this vasculopathy. Your vasculature inflames, you get internal hemorrhaging, and you die very quickly. And you can imagine if you're a human, there are no symptoms, you're just going to take the pill and die. So lots of reasons why you would really not want to target this, but lots of reasons in biology why you would. So how can we go at this? How can we do this differently than the rest of the pharma companies have done? So what we did is we stood back and said, okay, we're not going to be successful by targeting the catalytic domain. We have to somehow understand the regulation. If we understand how it's regulated, perhaps we can make molecules which target different forms of the enzyme, and if we do that, maybe we'll get lucky. If we're lucky, maybe if we try to understand how it's regulated, actually it's conserved, but if we understand how it's regulated with these different regulatory domains which are highly conserved between all PDE4s, and they're actually conserved across species. So in Drosophila and nematodes, you get PDE4 and they have these same regulatory domains. That's also important, right? Because if it's conserved, it tells you it's functional and it's important. And this UCR1 and 2 are unique to PDE4s. The other enzymes don't have that regulatory domain. They have different regulatory domains. The architecture is conserved, but its regulatory piece is unique. So already there might be a way that we can get at PDE4s but not the other. And if we're lucky, maybe we can get a PDE4D rather than the others. Something I didn't mention, the genetics and the biochemistry told us that to target for cognition and improve memory, we really wanted to inhibit PDE4D and not the others. So the first thing we did, we started looking at this, how are we going to do this? It became immediately obvious there's a real problem in the field and that all the enzyme assays for measuring PDE activity were horrible. They're endpoint assays. Basically, you have a chemical fluorescent probe which binds the phosphate, the product of the reaction, but doesn't bind the phosphodiester substrate. And you use this horrible fluorescent polarization assay, very low throughput, endpoint only. And so we came up with an enzyme couple assay to measure PD activity in real time. Basically, we coupled the phosphodiesterase activity to provide the other enzymes that you can follow. So there's adenylate kinase, pyruvate kinase, linked to lactate dehydrogenase. And as long as these are all very fast, which you can manipulate to make sure that they are, this is rate limiting. The conversion of NADH, which you can measure very easily fluorescently, can be measured in real time, linked to this. Every once in a while, you have to go back to make sure you're developing inhibitors that are actually inhibiting PD4 and not adenylate kinase, but that's easy to do. You get these great, easy to run assays, and so instead of doing single point time points in an Eppendorf tube, you can now do real time in 96 or 384 well plates. Get lots of really great data. There are actually error bars on these dots. We're doing it in triplicate, and you can see that the enzyme, the rate of the reaction is linear with a function of enzyme concentration. So with that in hand, one of the first things we did to characterize the assay, that never done in literature, it's amazing to me that it hasn't been done in literature, was meant to do a simple KM assignment. And we took the biological isoforms of the enzyme, PD4D or PD4B, and did this experiment. And Pam Whitty, who ran the experiment in the lab, great enzymologist, showed us the result. She was just lackadaisical about it, and I thought, this is amazing, this is fantastic. You have no idea what you just did. Because this opened up the field in an incredible way. Just the fact that the KMs were different, the active sites are totally identical, and yet somehow these enzymes with the regulatory domains treat the substrate differently. There are no inhibitors here, but it says for the first time, statistically significantly, a very different KM says that the enzyme must be handling the same substrate differently. There could be a handle, there might be a way that we can now get into the enzyme and make it so that it's for D and not for B. And it's remarkable to me that even when I went to management and lots of people, it was so hard to get anyone to appreciate this very simple result, but it was so critically important to the project. Next thing we did is we went to the literature, we went to the patent literature. With decades, pharma companies had gone through many, many different compounds. We reached out and synthesized or commercially obtained 58 different known PD phosphodiesterase inhibitors. The vast majority of them look like this, and we assayed them in our assay. So if you look at percent inhibition as a concentration of the inhibitor, and measured them against the catalytic domain or the catalytic domain with its regulatory piece there. The vast majority of inhibitors look like this. There's no difference. But it was exciting to us that we found one inhibitor that looked like this. It was actually one of the very first PDE4 inhibitors ever characterized. Roloperam has two stereoisomers, and that's why there's two lines here. And after that says a slight window. It actually does inhibit the long form of the enzyme, so with lower concentrations you get inhibition, whereas the catalytic domain you require higher concentrations. And there were two very special inhibitors. RS25344, which has been made by Roche, and PMNPQ, not clear where that molecule exactly came from, but those are both chemically related, and they had a very wide window. It wasn't appreciated that they actually had this feature, but they had a very wide window between the catalytic domain and the long form of the enzyme. That was pretty exciting. I said, wow, that might be a way in. So what do we do to understand function? You go after the structure. So we crystallized these things. They inhibited the catalytic domain, so we could take the catalytic domain, crystallize it with these different inhibitors. So here's roflumilast, and it's shown in yellow here. It sits in the active site. Hydrogen bond this conserved glutamine. It's held very tightly by hydrophobic interaction right here, reaches back, and it talks to the metals, which are important for catalysis, back in the active site. The next inhibitor is roloperam, and what we found with it was we can actually capture it in two distinct conformations in the crystal structure. In one case, it reached back to the metals, but in another conformation, it reached out here towards solvent. And that was exciting because the two that did have a big window, RS25344 and PMPQ, they had this unique feature that they actually sat in the active site and they reached out to solvent with these two critical phenyl rings. And it was when we knew from the other biochemistry, from the patent literature, that if you muck with these phenyl rings, you really affect the potency. So even though they're reaching out towards solvent, sitting back in the active site, they must somehow be involved in inhibiting the enzyme. So we came up with the hypothesis, maybe what's happening is that there's another domain which comes across the face of the active site, and interacts with this part of the molecule. And that's how these inhibitors are actually working. Seemed a reasonable idea. How do we get some confidence for that? Well, one of the experiments we did, when you look at this sequence, UCR1 and 2 stand for upstream conserved region 1 and upstream conserved region 2. They're conserved. There's not a lot of sequence variation. But there was one hydrophobic residue, which was not conserved across that, and that's a phenylalanine. And phenylalanines like to stack and interact in lots of different ways with those phenyl rings we saw in RS25344. So we had this experiment. Let's singly mutate every single phenylalanine all the way across the N-terminus of PDE4 and see what happens. And then what we did is we took those mutant enzymes and assayed them with RS25344 or PMAPQ. And then what I show on this graph is the IC50 of the mutant versus the IC50 of the wild type. Now you can imagine if the mutant has no effect, it's going to be an IC50 over an IC50, it's going to be 1. But if all of a sudden now one has a big effect on the other, we'll get a big change in the ratio. And of course, as you march along here, none of these mutations have any effect until you get to F196 or F201. Right down here at this end. So that gave us confidence. It told us how to make constructs. We actually went through 383 different constructs, all expressed in vector virus-infected insect cells, all purified to homogeneity, set up a little over almost a million crystallization trials. When we did it, we were able to crystallize this unique form of PDE4D and this similar form of PDE4B. So here's a catalytic domain with cyclic AMP sitting in the active site. There's this little piece of UCR2. Now, this linker here is very non-conserved, and that's not surprising. In the crystal structure, it's completely disordered. But sitting across this is this active site, and in PDE4B, because of the nature of the way the molecules pack and different crystallization conditions, you can see a little bit more of it. It looks bigger, but this is the exact same helix coming across the active site in both enzymes. When you look at detail on that, some really great things happen. So here is the RS25344. Here is PMMPQ, both in the active site. In this case, it's PDE4D. In this case, it's PDE4B. And you see these great interactions where, indeed, this phenylalanine of 196 is coming in and stacking in a pi-pi interaction. It's also making an edge-to-face interaction with the other phenylalanine of the drug. Phenylalanine 201 is making a critical interaction with the catalytic domain itself. We knew it was important, but it actually doesn't contact drug, but it is really critical for stabilizing the helix up against the enzyme surface. And what was really fantastic, I mean, if you look at this helix, it makes this really nice, it makes perfect biological sense. There's all these nice hydrophobic residues aligning with great shape complementarity from the UCR2 next to the catalytic domain. Also, if you look at down the helix, all these nice hydrophilic residues are pointing out towards solvent. So the whole structure feels good, it feels right? You have this nice antipathic helix, perfect shaped complementarity. But how do you know this is actually real? How do you know that you just haven't done something unique to the drug that traps this crystal structure? Because no one ever got this crystal structure, because the only way you can stabilize it is to have one of these ligands, one of these small molecules sitting in the active site holding it all together. Otherwise it's flopping around and it doesn't crystallize. So then we go back to our enzyme kinetics. I showed you this result before. Maybe this single phenylalanine to tyrosine difference that we saw in the crystal structure. In this case, there's a phenylalanine 196 in PDE4D, and all the other isoforms, A, B, C, and D, A, B, and C, is actually a tyrosine. And so we asked ourselves, could that be the source of the difference? And we did that because it actually made a lot of sense. We didn't crystallize this, but we modeled it. If you just take out the drug and superimpose and put in cyclic AMP, you now get this, and this tyrosine is in perfect position to hydrogen bond to the hydroxyl of the 3' hydroxyl of the cyclic AMP. And so we had this idea, is this the basis for the difference in the Km? And so what we did is we took the 4D enzyme, normally has a phenylalanine, mutate that phenylalanine to a tyrosine. And you can see what exactly happens. The 4D enzyme starts to look now exactly like a 4B enzyme. And that's cool because there's no ligand or anything in here. This is all in vitro, just with normal cyclic AMP measuring the kinetics of the reaction. There's no inhibitor, and yet exactly informed by the structure, you get the exact result that you'd expect. So that supplies a lot of evidence that indeed in vitro, the UCO2 is coming across and talking, and it's not just unique to this ligand, but the drugs we've discovered, these ligands we've discovered, actually bind it and stabilize it. So with that, we embarked on a couple years of chemistry, but I won't go into detail. I'll summarize in one slide. Fantastic group of medicinal chemists at Decode, the sister company of ours, Decode Chemistry in Chicago, led this effort. And basically what we did was we started to think about how can we move the binding energy away from the catalytic domain and move the binding energy to the UCR2. Because we wanted to have things which would now only bind and inhibit forms of the enzyme that have that UCR2. We also tried to make modifications which would exploit that phenylalanine tyrosine difference. And we were able to do that. And so earlier I showed you vast majority of the industry made these competitive inhibitors. This is going back to the catalytic domain versus the long form of the enzymes. This is where I showed you we started with roloprand. It has a little bit of window, PMMPQ, also had a nice window. The first generation compounds also had a pretty big window. As we evolved these things, made them better and better, we could get inhibitors that only inhibited PDE4D and would not inhibit the catalytic domain at all. What's also really critical is we can make enzymes which are very selective for the phenylalanine. One key modification is this fluorine modification right here. It creates steric and electrostatic repulsion with the tyrosine. It actually creates electrostatic attraction with the phenylalanine. A tyrosine ring has like this, and the electrons are going up and down, and as a result, you get a negative charge across the top of the phenylalanine. But across the side, the edge, you actually get a partial positive interaction. And so you put this negative fluorine right in space, trying to interact, so you get a big old negative fluorine with a positive edge of a phenylalanine, you actually get attraction with the drug molecule. And so that works great, and you can see you can get inhibitors which are very selective for PDE4D. So the IC50 is 8 nanomolar, PDE4B is 330. Quite a big window, and then you can do those mutagenesis experiments again. So if you take the 4D, mutate the phenylalanine to 196, you go from 8 to 280, just what you'd expect. And if you mutate the tyrosine in 4B, from tyrosine to phenylalanine, you go from 330 down to 12. Just what you'd expect. So now what we've done is we've created molecules which sit in the active site, sit there. What they can now do is reach out and grab a regulatory domain coming across the face of the enzyme and lock it in this inactive conformation. If we zoom in, when he does this, yep, you can see all kinds of unique interactions. So there's the fluorine interacting with this tyrosine. There's the 2-amol, which is also important for interaction with the regulatory or the catalytic domain. We have a nice stacking interaction here, critical hydrogen bond. And so we can make very potent inhibitors which lock this enzyme in this new conformation, which we couldn't, that all the active site inhibitors can't do. They muck up the active site, but they don't stabilize this really unique conformation with UCR2 across. So the question becomes, will those have any unique or interesting pharmacologies? So we started to test them in a variety of cognition models. So I'm only going to show you one here called the novel object recognition test. And so what you can do is if you take mice or rats, they're very curious animals, if you put two objects into a box and you let the mouse, or in this case a rat, explore it. So if you put two baseballs, it'll explore one baseball, it'll explore the other baseball, it'll become familiar with that baseball, and then if a little bit while later you put in a baseball and a golf ball, what do you think it's going to do? It knows the baseball. I've already explored that. I don't need to. I'm much more curious about this new golf ball that's in my cage. And it'll spend all its time looking at the novel object rather than the familiar object. But you can test memory by doing this experiment by varying the time. So if you first do this training test with two baseballs, and you wait just 1 hour, you can still discriminate just fine. If you then wait 3 hours, you start to lose a little bit because the rat's forgetting what the familiar object was. And after 24 hours, it completely forgot what a baseball looks like. And so it spends just as much time with the baseball as it does with the golf ball. So now what we do the same experiment with drug. And so in the absence of drug, 24 hours after the training, the rat can't discriminate. But if you give it rolopram, a known molecule all the way back from the 80s that improved memory, you can see now you start to discriminate. Now we give it our drug in a dose-limiting, escalating sense, and you can see that as you give more and more drug, the memory comes back. We did it with two different kinds of compounds. What you can hopefully see is that the medium effective dose is, in this compound, is 30 micrograms per kilogram. And look at rolopram, it takes significantly more, it takes 100 micrograms per kilogram. What the side effects, remember rolopram had these really horrible side effects. One of the great species to study vomiting, it turns out that mice and rats, I didn't know this, but they physically, literally cannot vomit. So you can't study that side effect in a mouse or a rat. What you do is you go to these really horrible creatures, these Asian house shrews that are easy to house, really nasty animals, but they vomit like crazy. And they throw up. And so if you feed them and you give them a little bit of rolopram, it doesn't take very long before all the shrews in the cage start throwing up. But you can give significantly higher doses of our compound before you see any emesis. And we know that in this control experiment, you're still getting this compound into the brain and through the animal, but it doesn't cause it to throw up. And so now we have made compounds which are more potent in the memory assay and significantly less effective in the bad toxic or emesis assay. So we've proven up a huge therapeutic window. And depending on the species, it's anywhere from 500 to 10,000-fold greater for our new allosteric modulators than for traditional competitive inhibitor. There's also this really nice feature around vasculopathy. It's a hard thing to assay. This stuff that I told you about where the rats really hemorrhage internally. There are biomarkers. One of them is TIMP1, and so if you give just a little bit of roflumilast, one of those active site inhibitors, to a rat, right away, as soon as you give it, the TIMP1 jumps way up. And for ethical reasons, we stop the experiment right away because we know that's going to be very bad for that mouse or rat. But our compounds, you can see that you can go to significantly higher concentrations and absolutely no induction of any of those biomarkers. And through a variety of histology, we show there's no indication of any vasculopathy in these rats. So we've bypassed all the bad effects by making these new kinds of inhibitors. So right now, those kinds of compounds are being developed by a company called Tetra Discovery. They're a great company based in Michigan. The work I told you about was published in 2010. But we're continuing that work. We still collaborate and work with Tetra. Mark Gerdes, the CEO, does a fantastic job. He's actually now been able to make inhibitors that are not only selective for PDE4D, but are also selective for PDE4B. And they're going after indications including not just cognition and schizophrenia, but traumatic brain injury, and a variety of other things that's really exciting. And then there's also a brand new paper reproducing the crystal structure from Pfizer. Also, Pfizer is now interested in these compounds, and they're continuing this effort. So that's the kind of story. But I wanted to just bypass and give a little bit of a soapbox here, because I have the opportunity. It's like, we should have known this. It seemed like that was a really hard thing to do. I told you it sort of took us 3 or 4 years to go from the idea to this idea to actual molecules that do this. We should have known, in fact, the industry decades ago should have done this because there are lots of molecules which already do this. I'm going to skip this and get to propelled A. This is a natural product. This is made by a microbe to fight fungi, and it does this exact sort of thing. It sits on a protein surface and stabilizes a protein-protein interaction. This is a G protein, and G proteins are notoriously difficult to drug. In fact, the industry has been trying to drug KRAS, the most commonly mutated G protein, most commonly protein in most human cancers. And what this molecule does is it tricks. It binds away from the active site, the enzyme binds GTP way too tightly, so you can't get it off. What it does is it binds over here, stabilizes an interaction with another protein called SEC7, and holds it in an inactive conformation, exactly like what our B4 allosteric modulators do. Then it comes in here and makes stabilized interactions between both the green, the G protein, and the cyan, the SEC7 protein. There are multiple conformations. It stabilizes not only interactions directly as a small molecule, but between the protein surfaces. And so we should have known. I mean, nature's already shown us this is how you can drug really difficult protein targets. Another known example that's been unveiled very recently is ubiquitin and auxin. Auxin is another molecule that sits right here and it stabilizes an interaction between ubiquitin E3 substrate adapter and this is a repressor, and it stabilizes that interaction. When that stabilization occurs, you ubiquitinate the repressor, it degrades the repressor, and so then you can turn on transcription down here. What normally happens in the absence of auxin is this inhibitor binds your transcriptional factor, inhibits it, and what auxin is really doing is specifically stabilizing an interaction between the repressor and the ubiquitin ligase, targeting it for degradation, so that now you turn on transcription in the presence of auxin. That's exactly what our compounds are doing too. They're stabilizing the protein interaction and having a result. People have been trying for decades to target transcription factors, but you can't do it. There's no enzyme active site, so the industry thinks, well, it's undruggable. It's on an active site, I can't do it. But this actually shows you this is exactly how you can do it. And in fact, very recently, the Broad Institute has been doing some great studies with a known drug called lenalidomide. It's a derivative of phthalimide. It's been shown to be very effective in multiple myeloma. And it uses this exact same reaction in human cells. It stabilizes an interaction between cereblon, the substrate adapter, and IKZ transcription factors, IKZ1 and 3. It targets them for degradation. So then now you get a whole series of transcriptional outputs. Most importantly, you downregulate MYC, one of the most important transcription factors known for cancer, and you can cure or affect multiple myeloma. And so once again, nature is showing us this is the way you get undruggable targets. You don't go after the active site. You go after stabilizing a protein-protein interaction, which locks it in an ineffective conformation. So that's my little bit of soapbox. And I will stop there because I think I've been going long enough.
Hey Alex, what year was the KM study done for the two?
The KM studies were done around 2005 or 6.
I can't believe it.
I know, can you believe
I
this?
just can't believe it wasn't done before.
And there was no good assay. They realized they had this. I mean, the problem is that the substrate and the product were so similar to each other. That it's not an easy way to.
Yeah, remarkable.
But we knew at the beginning of the project that having a good, reliable assay was actually going to be absolutely essential. I didn't really get into it, but it turns out PDE4 is a dimer. We have these really complex models which are all completely consistent. The new structure from Pfizer shows that PDE4 is a dimer in solution that explains all the kinetics that we saw. I didn't get a chance to go into that, but it was absolutely essential. We had a good kinetic models and understanding of the kinetics before we even started the project. So a few people, I think, appreciate how laying that groundwork is so important before you start to try to make drugs.
Why is TIP1 a biomarker?
I can't tell you why, because the vasculopathy is such a uncharacterized, unknown process. But the drug companies went off looking for biomarkers to screen their own inhibitors. And so it was known in the literature that it was a biomarker, but I don't know more than that. There's several others, but that's one that's really easy to assay.
What's the mechanism of the improvement of dementia? Is it through a vascular mechanism?
So, it's pretty clear that memory is in part a result of making connections, lasting connections, synaptic connections in the brain. It's a intracellular result. And to make those long-lasting connections, you have to have a result of transcription and translation in the two neurons. And so two neurons that interact with each other, you can have an effect through CREB, CBP, and it's not completely understood exactly how this happens. But the idea is that in the brain there are signals when neurons start talking to each other. PK is one of the critical enzymes which phosphorylates CREB, and then that leads to activation, and that's a good thing for memory. But there's also active phosphodiesterase in the brain, which is degrading and inactivating the PKA, which in turn results in decreased phosphorylation of CREB, which results in a decrease in the transcription and translation at the neuron. And so by inhibiting this, you actually get more and more transcription, and those connections between the neurons become more long-lasting. That's the idea that's not completely understood.
Okay, Alex, do one more.
Yeah, so I was curious, when you switch the binding energy over to the regulatory domain, does that sort of open doors for other conserved regulatory domains, or is this a pretty unique domain?
Great question. So it turns out I glossed over this, but it turns out that there are actually two different regulatory domains. The one I showed you was UCR2. There's another one at the C terminus. It wasn't even appreciated as a regulatory domain, and we've named it CR3, control region 3, because unfortunately it's not at all conserved with UCR1 or UCR2, but it actually can also come across the active site. And sequence differences in that one make it easier to make the 4D selective compounds. And so the reference that I showed you has all been using a completely different regulatory domain. Also, results of the Burn Institute, there's another group looking at PDE3 in cancer. They also have molecules, they haven't gotten the crystal structure yet, but they also have molecules that looks like in PDE3 are reaching out and talking to a unique domain. But I think it's going to be, again, it should be obvious. It's conserved. That regulatory mechanism, if that architecture is conserved, that regulatory mechanism must be conserved. And so if we can do it for PDE4, we should be able to do it for PDE3.
What other, well, as you originally said, nature will teach you something. What are the other allosteric modifiers naturally existing? Because obviously,
Right. So I have a hypothesis that I didn't have the luxury of exploring at the company, that there are actually metabolites doing this in our own body, and that there must be molecules sit in the active site and do exactly this. I think in the cereblon story also, I think it is not coincidence that lenalidomide does this exact same thing that auxin does. I think there are metabolites which are regulating enzyme function in the body. We just don't appreciate what those metabolites are. We did do a little bit of study trying to find small molecules, we fractionated cells, tried to find something that would do it, but in a short effort we were unsuccessful.
Yeah, just quickly, would you imagine that those metabolites might come from microbes that are
Of course. That's very likely.
Or is that too risky a strategy to put something that's important, not in the human?
That would be courageous. No, I think that I wouldn't be surprised, right, that if we're making metabolites, after the session this morning, that are going to the gut and modulating bacterial biology. It's sort of an obvious way. I mean, now that you see it, it wasn't obvious to me 10 years ago, it was obvious to me now. Of course, it's conserved. If nature has come up with that solution, that's a very easy way to do it.
Alex, thank you so much.